Okay. Are you ready, Carol?
Yeah.
Okay. Okay, we're going to get started. I have a cold, so I may not be the most coherent person on the planet. I'm Shannon McQueenie. I'm the division head for bioinformatics and computational biology. Dr. Hirsch is away, and so I'm going to do the introductions in his absence. It's my pleasure today to introduce Melissa Yan. She's going to be presenting work for her master's thesis proposal. Some important things to remember: this is a great opportunity for our students to present their research ideas and get input. If you ask questions, we ask that you remember that we're recording, and this is being streamed, so just hold the button down so that the question actually gets captured. When you ask the question, keep it held down. If you're watching this on the web, and you have questions you would like us to ask her, you can tweet them. If you use the hashtag DMICEConf, C-O-N-F, you can barely hear me, DMICECONF, or tweet it to OHSU Informatics. I'll be monitoring that, and we can ask her at the end. Melissa is in a program that's a partnership with Portland State University, in which the students start off on kind of an accelerated bachelor's in computer science and then come here to finish. So it's basically like a very expedited 3-year bachelor's program, and then come here for their final 2 years for the master's in biomedical informatics. And with that, Melissa, the floor is yours. Thank you.
All right. So can everybody hear me okay? Okay. All right. So today my talk is implementation of the HotNet2 Network Diffusion-based Analysis Method in ReactomeFIViz. So an overview of today's talk will be an introduction to networks, the HotNet2 algorithm, current implementations, and my specific aims for this project. So high throughput experiments produce a lot of complex data sets. These data sets are readily available to the public, but this data often contains only partial omics information, and it's often very noisy and contains a lot of false positives or negatives. So a big issue we have is how can we process, extract, and explain the data to better understand diseases and biological processes. So a popular method is to combine biological data with networks and then extract and explain patterns in biology. So this has already been done in predicting protein function, comprehending chemical reactions in organisms, and detecting relationships between diseases. Computational tools are able to do these things because networks can be represented in the form of an adjacency matrix. So using these matrices, computational tools can analyze data, detect patterns, and integrate additional biological data. To understand why it's important to use networks nowadays, I'm going to provide a little bit of background on how interpreting biological data has changed. So in the past, we take a complex data system and break it down into smaller, simpler, basic individual components to study. So one gene for one disease. And although this was beneficial in the past, most studies now, they show that some diseases are caused by multiple mutated genes. So now we're more focused on a holistic approach where we study multiple components within and between complex systems of genes, proteins, cells, organs, and organisms. An example is where we inspect multiple mutated genes for a single disease, how those multiple mutated genes might be related to each other, and the possibility that they might also impact other diseases. So this provides us with a systems perspective, and using networks, we can integrate different levels of information to help us better understand biological systems and diseases. So in regards to disease studies, we can map these diseases onto a protein-protein interaction network, which is shown in gray on the screen. So the circles are nodes, and these represent proteins or genes. And the lines are edges. These represent functional and/or physical relationships between genes. So if we're interested in studying the relationship between two different diseases, shown in green and purple, we can map these diseases as a group or module onto the network. So as you can see, there's two genes that both have green and purple, and that just means that those genes overlap in both disease A and in disease B. So furthermore, networks can also be used as models to apply concepts and algorithms to biology. So a concept I'll be talking about is heat diffusion. So if you have a source of heat, that heat can disperse into the surroundings that are cooler. So to illustrate this, when we're given a network with a hot central node, before diffusion, that heat will stay in the same place. But as diffusion occurs, that heat in the middle will slowly spread out and heat up the surrounding cooler neighbors. And so this is what will happen. So this concept of heat diffusion, we can apply it into biology. So here's an example. This is a KEGG pathway map, and specifically it shows the JNK and P38 kinase pathway. It's involved with cellular functions such as proliferation, differentiation, inflammation, and apoptosis. So if JNK were to become mutated, it will block the upstream signals from the genes shown in pink, and that will impact the downstream signal to the genes shown in blue. And this will cause cellular function changes, and it will also affect the P53 signaling pathway. And so this just goes to show that when one gene gets mutated, it can affect multiple genes throughout the network. And by using diffusion, it's possible to model this functional impact of how genes will impact each other across a biological network. So to use this idea of diffusion, we can combine it with network data and gene scores to observe how the individual genes will influence each other in a network. So the network represents how genes are related to each other physically and or functionally. And gene scores are a list of genes where each gene is assigned a significance value based on the study of interest. So an example is for disease studies, one of the main goals is to find key candidate disease genes. And the network we commonly use is the human protein-protein interaction network. And the gene scores are a list of genes, each assigned a significance score based on their potential involvement in a disease. And that significance score can be based on p-values or rankings from previous studies. So going into a bit more detail about the gene scores. So the gene score significance can be converted into a heat score for heat diffusion model. So the more significant a gene is, when it's converted, it will have a higher temperature when converted into heat. And so this enables us to use specific gene information and combine it with the network data. And in biology, we already know that genes will impact one another. So if we're given genes of varying significance level in the form of gene scores, then we can place that onto the network as a model, and we can get a better understanding of how significant disease-associated genes will affect or influence other genes. Because the significant genes will diffuse their scores into the surrounding neighbors. And so this approach of using gene scores and networks is known as the diffusion-based approach. An algorithm that uses the network diffusion-based approach is HotNet2. And so there are 4 steps in HotNet2, and I'll go over each of these steps in detail next. So in step 1, we use diffusion to estimate how genes impact each other. So to determine how all genes will impact one another, HotNet2 uses a diffusion process known as insulated heat diffusion. That means that in the process of diffusing heat from neighbor to neighbor, each of the nodes will retain some of its own heat. And this is determined by the beta parameter, which represents a fraction of the heat retained. So in this example, each node will retain half of its heat, and that means the remaining half will be shared equally among its neighbors. And so given a pair of genes A and B, we're going to determine how heat is transferred. So from A to B, A only has one neighbor, so A will retain half the heat, and the remaining half will be transferred into B. And from B to A, node B has 5 neighbors, so node B will retain half the heat, and the remaining half is shared equally among those 5 neighbors. And so the gene-to-gene relationship from A to B is half the heat it will get transferred, and from B to A, only a tenth of the heat gets transferred. And then this step is repeated for all genes within the network, so we can get an impact estimation of how all genes will impact one another. And once we have an estimation, then we need to determine the actual impact genes will have on each other. So to do this, we combine the results we obtained in step 1, where we estimated how much heat was transferred, with gene scores. And these gene scores are, as a reminder, they're significant scores. So these significant scores are then placed onto the network, like so. So for A, it's 0.01, and for B, the significant score is 0.02. And then we perform matrix multiplication by multiplying the gene scores with the estimated impact for all genes. And then, so the actual amount of heat transfer from A to B, estimated it was half, but in reality, it will be 0.005. And from B to A, estimated was 1/10, but actually it will be 0.002. So based on the actual amount of heat exchanged, we will now have a network containing information about the significance of each individual gene and the amount of heat that is exchanged between all the gene pairs in the network. So using this, it brings us to step 3, where we can obtain subnetworks by removing gene relationships below a threshold. So to illustrate this, let's look at the legend I have on the screen for heat transfer. So the red arrow means that high amounts of heat are transferred between nodes, and the white arrow represents less amount of heat is transferred. So in order to extract these subnetworks, we need to set a threshold to remove gene-to-gene relationships. So if we set the threshold below that orange arrow, it will filter out the yellow and white heat transfer arrows from the network. So here's an example after filtering. As you can see, based on that threshold, only the red and orange heat transfer arrows are left, and the yellow and white arrows have been removed. So after that threshold, we can group the remaining nodes into subnetworks, like so. And after this, we move to the final step where we determine if these subnetworks are significant or not. So in the final step of HotNet, we determine the significance of the subnetworks by evaluating the p-value and getting the false discovery rate. So this means that for all possible subnetwork sizes, p-values are obtained based on the number of subnetworks for a given size in the actual network, and then it's compared against the randomly formed networks. So continuing the previous example, in the previous example, we'll obtain this actual data table. And so this means that for a subnetwork of size 5 nodes, we see that there's one subnetwork that's circled. And for a subnetwork size of 3 nodes, there's also one subnetwork that is circled. And then in the network, we create the random networks by using the same nodes but with different edges. We rerun HotNet2, and then we will create a similar table to this. And by doing so, we can compare the subnetworks in the actual data with subnetworks in the randomized data to get that p-value, which will indicate how significant subnetworks of a certain size or greater are. And then with those p-values, we can calculate the false discovery rate. And so the next slide, I'm going to demonstrate how to get those p-values in case my description was a bit confusing. So continuing from the previous example, we have the actual data set. And then for the random data sets, you either perform hundreds or thousands of permutations. And for the sake of this example, I'm only showing 2 of the 100 possible random data sets. So in the actual data set, we can set a threshold to be 3 nodes. And then we need to also set that same threshold in the randomized data sets. And then we can plot the distribution of and get that p-value to test how significant subnetwork sizes greater than or equal to 3 nodes are. So using p-values, we can then calculate the false discovery rate for the subnetworks that we find. And by completing these 4 steps, HotNet2 is able to integrate network data and individual gene information to find subnetworks that are potentially involved in diseases. So from the Liaison et al. 2015 study, they're the ones that came up with this HotNet2 algorithm. They were able to detect the SWE/SNF complex, and this is a known important tumor suppressor subnetwork. And so this is what it looks like. And if we want to check the frequency of mutated genes in patient tumors, because their gene scores are based on the frequency of mutations in patient tumors, so we want to map this subnetwork back to see something important. So when we map it back into that frequency thing, the hot genes are the ones that are highly mutated, and they are also possible known cancer-causing genes. And the rarely mutated genes are the cold blue ones, and they're considered insignificant. So as we can see in that significant subnetwork, it includes one of the blue genes, ADNP, and that means it has a low frequency of mutating in patient tumors. And so this subnetwork just shows that frequently mutated genes and rarely mutated genes, when you combine them together, they can actually play a role in cancer and are important. So using HotNet2, subnetworks of known mutations can be expanded to include interacting genes and proteins that might also be associated with cancer but are usually ignored because statistical tests deem them as insignificant. And so for those interested in using HotNet2, there is a current implementation available in Python, but you need command line software scripts to run it, and it's also very time consuming. And because you have to use these command line scripts, it's probably difficult and unintuitive for biologists or clinicians without programming experience. And to the best of my knowledge, there's known software that provides a graphical user interface for network visualization analysis of the whole HotNet2 diffusion algorithm workflow to users. So to make HotNet2 more intuitive, I'm going to implement it into Cytoscape's ReactomeFIViz app. So Cytoscape is one of the most popular biological network analysis and visualization tools. It's a platform that's Java-based, and it has about 150 apps that can assist in network analysis, and ReactomeFIViz is one of those many apps. So ReactomeFIViz performs network visualization analysis using the Reactome functional interaction network of biological pathways, and it contains about 60% of the total human genes. So I chose ReactomeFIViz because it has already implemented a previous version of HotNet2. So this means that my project, I can use that previous implementation as a framework to build upon, and I can focus on optimizing HotNet2 and providing users an intuitive network visualization tool. So because ReactomeFIViz and Cytoscape are Java-based, my project, I'll implement HotNet2 in Java, and based on the results on the screen, we expect that there will be performance gains. So to solve issues related to making HotNet2 more intuitive and optimized for visual analysis, I propose the following objectives. So I want to implement HotNet2 into ReactomeFIViz, a Cytoscape app, and for the first time, provide biologists and clinicians a tool to analyze their own data using this powerful diffusion-based algorithm. And they can also use this algorithm alongside other pre-existing network and pathway features using a suite of rich graphical user interfaces. To accomplish this objective, I propose the following specific aims. So in aim 1, I will port HotNet2 from Python into Java. And the method I use is this. So I'll implement HotNet2 into Java and compare the results with the Python implementation to ensure that my results are correct. And the assessment will be based on accuracy, performance time, and memory consumption. So by following this procedure, the finalized version of code will be correct, robust, efficient, and usable for future integration into ReactomeFIViz. To assist with code assessment, I'll use the visual VM profiling tool that's provided by Java. And this just tracks performance and memory usage to inform programmers which methods they should improve upon. So for performance information, it provides you a picture and information on the method's execution time and the number of invocations that are made. So this just provides insight to me on which functions I should probably focus on and optimize to decrease the execution time and the number of frequency of method calls. And for memory usage, it can be used to detect unnecessary memory usage, memory leaks, and bottlenecks. So by understanding where and how memory is used throughout the development process, code can be better optimized to handle large data sets and prevent issues from occurring. So since HotNet2 uses the human interactome, it's a very large data set, and it also needs to perform hundreds to thousands of random permutations to get the results. So it's very ideal that my code is able to perform quickly and accurately. So it's very essential that I track and make sure I'm using the correct data structures and my functions are correctly implemented, because this will assist in minimizing performance time and minimize memory consumption. So once the code is optimized, I'll begin aim 2, where we integrate HotNet2 into ReactomeFIViz as a GUI-based intuitive network analysis tool and evaluate the usability among researchers. So the approach used is first I'll integrate Java version of HotNet2 into ReactomeFIViz using the previous version as guidance. And then after integration, I will modify and add visualization features related to the input format, network visualization, and parameter selection. And then finally, a usability test will be conducted to ensure that these features actually do assist in analyzing data. So here's a workflow of what the user will be able to do through the workflow. So the input, they can provide a mutation annotation file or gene scores. And while HotNet2 is running, they'll be able to view the progress to see how much time is left before the job is completed. And the results presented to the user will be a list of significant subnetworks and visualized subnetworks. So for network visualization, the user can view the nodes colored according to their significance. So hot means most significant and cold means least significant. And this just provides user details on specific individual genes within the network. Or they can choose to view the network in the form of colored subnetworks. And so it's expected that the GUI workflow and the network visualization features will enable researchers to analyze significant subnetworks more easily. So to ensure that these expectations of mine are met, ReactomeFIViz with this HotNet2 algorithm and these new features will be provided to users. For user testing, users can use their own data or provide sample data or use the provided sample data set. And prior to testing, I'll only provide users the ReactomeFIViz tutorial from reactome.org, and that's just to get them familiar with the pre-existing ReactomeFIViz features. The tutorial does not contain any information on how to use HotNet2. So following testing, the users will be asked to fill out a systems usability scale questionnaire to assess if HotNet2 and the visualization features are useful or not. Questions include ease of access, issues with usage, and appearance, and I'll also get additional user feedback to improve the HotNet2 integration before my project is complete. So in the usability testing questionnaire, participants are asked to score 10 questions on a scale from 1 to 5. 1 is strongly disagree, and 5 is strongly agree. The score will be added, and then it will be ranked based on a percentile to determine if the usability features were helpful or not. So upon completion of specific aims 2, users will be able to easily analyze data using ReactomeFIViz's implementation of HotNet2 with new GUI features. So in conclusion, the proposed project will extend ReactomeFIViz by incorporating HotNet2 as an interactive network diffusion-based analysis feature for biologists and clinicians. The user-friendly software with this optimized algorithm will accelerate disease gene analyses to benefit biologists and clinicians with and without programming experience. And users can also take advantage of Cytoscape's about 150 apps to further enhance their analyses for further research. And so I would like to end by acknowledging Dr. Wu as being my advisor, thanking Dr. McQueenie and Dr. Wilmot for being my committee members. And I'd also like to thank the DMI staff, in particular Ms. Diane and Ms. Lynn. I'd also like to thank my family and my friends in the VCE cohort. And I know I have some extra time, so before I go into questions, I just want to describe the previous version of HotNet2, in case you guys were wondering. So the previous version of HotNet2 is called HotNet, and there's issues because it's very biased towards these star-shaped clusters, like the one you see on the screen. So that middle node will be very hot, and it'll be surrounded by a lot of cold nodes. And so to overcome this issue, HotNet2 was developed, and it uses a different type of heat diffusion. So in HotNet, you use heat diffusion where all the heat in one node is transferred to the other. And in HotNet2, you use insulated heat diffusion, so each node retains part of the heat and the remaining half is shared. HotNet ignores the direction of heat travel, so from A to B, it will choose the least amount of heat that gets transferred and record it for both. But in HotNet2, it records the heat separately. So from A to B, it will have its own score, and from B to A, it will have its own heat transfer as well. So to illustrate this, for the HotNet input, if we give it a star-shaped cluster, you can see how for HotNet, it doesn't have those arrows giving heat directionality. And so the output will be that star-shaped cluster. But in HotNet2, it includes directionality. And so the cold genes will be removed because only the hot center node is there. And then another example, it's not the star-shaped cluster anymore, but we can see how HotNet, it doesn't remove any of the cold genes. And in HotNet2, because of W and S, there's heat transferring between both of them. That's more significant than just the hot S node transferring minimal heat to the T gene. And so, thank you. And any questions?
Questions for Melissa?
It's about your evaluation. Where are you going to get your subjects, and do you need an IRB protocol for this?
Yes, I do need IRB, and so I'm working on that right now.
Who are you going to use as subjects, or how are you going to recruit your subjects for this?
So I'm thinking about professors in the department, and if they're not available because they're probably really busy, then people from the BCB cohort.
I don't know, maybe if there's a way to get, like, the HotNet user community and recruit people from that, wider scope that would be, less, I don't want to say biased, but less well known to you, so maybe more optimal.
Yeah, that's a good idea.
Actually, can I follow up on your question? So actually, I want you to think about that a little bit, though, because if you have people who are very familiar with, there's two pieces that you might end up evaluating. And I looked at your questions, you have to think about this. One would be, are they evaluating how easy it is to use the implementation? They know HotNet, and then Aaron is thinking about this more and more, and then Aaron's dead on. But your pitch is for biologists and like clinicians and people who wouldn't necessarily be experts in HotNet. And if that's the case, it's actually a different evaluation. Yeah, right. And so it's really important to think about that, because then you wouldn't, many people in the department may not be familiar with this analysis, and especially from the clinical faculty, I would argue that's probably true. But you probably would like to get some of the, especially from the biology sides, from some of the biology departments. But you do need to think about that, because like the question you had up on there, Melissa, if they're saying how easy is the system, well, it could be super easy to run it, but if I don't understand the algorithm, so you want to make sure that your questions differentiate between that, or that your user base does.
So, there's a couple of user bases and a couple kind of questions you could look at this. You could look at this as, the current implementation versus the old for users that are familiar with this, and what's the difference or the improvement. Or you could look at people that are not familiar with using it but would be interested in using it and would have data that they would find useful to apply to this. And just, de novo, how would they, how did they find using this? And then you just straight use the system usability scale. But certainly you'd want to pick a set of users that you think would, in their normal work, benefit from this, even if they're unaware of it right now.
I have a couple questions about porting the Python code over to Java. So are there packages that are used by HotNet2 that you would have to worry about rewriting in Java, first of all?
Well, the paper for HotNet2 is very nice, and they have an algorithm for it, and so I'm kind of just following the matrix multiplication using Java libraries instead of NumPy for what the Python implementation used.
And then since there's a random component, there's a bunch of permutations. Is there a way to generate the same random numbers between Python and Java so that you can check that you're getting the exact same result?
There's a parameter you set when you're running the program, and so I'm not sure if it'll be the same networks, but I can definitely feed my program these networks to ensure that they're the same. Is that what you're asking?
No, I think what he's getting at is that so the depending on how they implemented the randomization and the way that they're using the seed, you'll see there'll be stutter. There'll be variation between it. So that's something that you need to think about in your implementation. Is that if all the parameters are the same and the seed is the same, you get the same result.
And and the last one I had is if they release updates for HotNet2, they find some bug in their code, do you plan on trying
then
to
I
follow
would
those?
plan on updating this as well.
So kind of going back to that last point, I think it's really important to think about contributing, if you are going to do contributing this back and making sure it's on Git and making sure people can access your code and that they can, so basically giving it back to the community to develop it. Because the nice thing about you doing this is that Cytoscape in particular has been active in very large development communities, but that's really essential to get that code back out there and can be used.
Yeah, I was going to ask about that. Is there a particular way that Cytoscape add-ons are distributed and maintained and made open source and been made part of the community, or is it just that there's sort of a collection of projects? Some of them live on GitHub and some of them live someplace else. Is there a formal way to make this part of the Cytoscape environment?
So Cytoscape, it's just a platform, and you just easily click and install these plugins. So these apps, whatever you're interested in, you just easily click, and then it's installed, and you can use it.
But he's getting at how it's managed. So that's how the user does it. What he's getting at is the way that Cytoscape presents it and how it's managed, how the packages are managed, what you're contributing to. Do you remember that?
I'm not sure.
Cytoscape has an app store, so basically you can install and do update.
It should be contributing.
Yeah, so our ReactomeFIViz is one of the But they don't manage all this source code, so each project can manage their own source code.
Okay.
Yeah, we have a public GitHub repository already set up.
So how does it get from GitHub to the App Store?
That should be manual update.
So somebody's going to have to own and manage this over the long term, or it's going to become not available to users. It sounds like. If there, it sounds like there's you could put it on GitHub, but then you'd have to build up a developer community that was always, moving the latest into the store or something.
I think one thing to remember, which she touched on in the beginning, is that she's developing into a plugin that already exists and is maintained by another group, which is ReactomeFIViz. So she's making a module for that. I think it's really important for our students and our faculty to think about that if you do it on your own, all of it rests with you. But if you're contributing into a larger project, you can leverage.
A somewhat general question building off of that is, how do you retain intellectual property in a setting like this and still make it make it available and shared?
Well, my intent was to provide it on GitHub and share it, because this is not my algorithm. This is written by another group, the Raphael's group at Brown University, and they have open sourced this algorithm, and it's on GitHub. And so I'm just taking what they have done, putting it in a new setting that's easier and more intuitive to use. And so people that use Cytoscape and are interested, they can just use the ReactomeFIViz app for it.
I think his question is bigger than that, though. I think his question is, so in this case, everything you're doing is open source. The algorithm was open source. There's no original art. I think the question would be, how do you communicate, and this is a bigger question than maybe you, but it's a good question. How do you contribute when it is your algorithm so that other people can use it, so that it can be incorporated and still retain the IP? And I'm not sure that she'll have you. We can take a stab at it, but that's a big question.
I'm not sure, but I would assume that I have to get a paper published on it with results, and then show people this is my algorithm, these are the results, and it's actually useful. And then so I have my name kind of associated with the algorithm, but then it's still available for people to use, kind of thing, right?
So what he's getting at, though, is that the publication is still separate than intellectual property.
Oh, okay.
So it is something to think about, and I know half of the students and postdocs were in a meeting who discussed this with tech transfer, and they had a symposium, but it's a really important point that you do need to consider, especially for novel algorithm development. You do need to consider and have a conversation here with tech transfer and business development about this in terms of before you even publish, just really to determine what is the best route. And that nothing precludes you from sharing it and making it open source. It's just that there's a lot of different licenses and they have a lot of implications.
Yeah, thanks, Shannon. I'm really, I do believe that the open source and shared policies that we're moving towards compared to where we were many years ago in many areas is great. It's also important to consider that the work that you're going to be doing in your careers, and I'll look around the whole room, you're going to be developing processes that it will be important for your careers over time to make sure that you retain certain rights at the same time, and to think about that and to learn about that process over the next couple of years during your academic training.
Have you ever and this is like an unanswered question, and having played with a previous version of HotNet, have you thought about how to integrate multiple data types, for example, including mutation and expression, in an analysis for HotNet?
I haven't, but I know there is an algorithm that already has that. It'll incorporate mutation information and gene expression information. And so that's called the tie-dye algorithm, if you want to look into that, because I'm still unfamiliar with it.
You mean using the same? I think he's asking in a diffusion framework. So you're saying, is that one that's doing it in diffusion?
Yeah, tie-dye also does diffusion.
So the one thing that really kind of occurred to me is that a real opportunity here, and this is might be beyond the scope of your current work, but is definitely kind of thinking about giving people the big picture of the integrated data, because not only is the expression important, but also epigenomics and mutations and copy number analysis. So if you could kind of figure out a way to kind of include all of that data and give people a big picture view of how that interacts, it would be really powerful.
Okay.
Simple way how you just run HotNet several times. You have either different data types.
But you still wouldn't get the integration when he's getting at it.
Some kind.
All right. Any more questions for Melissa? If not, please join me in thanking her once again.